Hello, today we're going to discuss the 1.5 notes on the cell theory. Now, we've already talked about one of the characteristics of life is that all living things are made out of cells. So the cell theory incorporates some of the important information that we know about cells. Now remember, we've discussed earlier, a theory is an idea that is backed up by evidence. So when we talk about the cell theory, we're talking about all the evidence that we know of that supports the idea that living things are made out of cells and how those cells actually work. Part of what I want to talk about today is some of the people that are involved in formation of the cell theory. It's often useful to answer the question, how do we know this stuff? It's not that a scientist wakes up one day and says, you know what, I think cells. In fact, it's a collaboration of many scientists all contributing little pieces of information and then collecting that information into one large cohesive theory. The first scientist we want to look at is Anton van Leeuwenhoek. He was a Dutch physician that was very interested in studying the world. And at this time, during the early 1600s, the microscope was this new invention that was all the rage amongst people that were interested in science. And Leeuwenhoek didn't want to just go out and buy a microscope like some of the other fancy scientists were. He wanted to build his own. So Leeuwenhoek actually designed several different models of microscopes that worked in various ways. But what Leeuwenhoek was able to do with his designed microscopes was he was able to view things on a much smaller scale that people had not seen before. He was one of the first people to kind of unlock this microscopic world that surrounds us. This picture here shows the design of the microscope that he invented, or at least one of the designs he invented. And it's very different from the microscopes we recognize today, but it had a lens and it was able to magnify objects to appear much larger. As a result, he was able to see things that people were not able to see before. One of the important discoveries that Leeuwenhoek made while looking underneath this microscope is he saw for the first time microscopic organisms. That is, organisms that are smaller than the eye can see. So this was one of the first times when people realized that life could be very, very small. And this really changed the way that we viewed life and then viewed other factors as well, like medicine. Long before Leeuwenhoek came along, our ideas of why people got sick had a lot to do with supernatural explanations, like evil spirits or the phases of the moon. But now people started to realize, well, maybe these little organisms are causing trouble inside of our bodies. Now, at the time when Leeuwenhoek found these things, there was no name for what to describe these as. So he called them animalcules. So if you imagine taking the word animal and the word molecule and smashing it together, you get animalcule, which literally translates to little animals. Now, following in the footsteps of Leeuwenhoek at about the same time, in about the mid-1600s, another scientist was using microscopes to examine various things and looking at the microscopic world. His name was Robert Hooke. He was an English physicist, and he was not as much interested in biology as he was in the physics of magnification. So he was more interested in what the microscope could do rather than what he was actually staring at, although he did make several important discoveries through his microscopes. One of the things that Hooke did was he took a slice of cork and he put it underneath his microscope. And what he saw were tiny little boxes, basically little squares. And he said to himself, this kind of reminds me of the cells in a monastery. So the cell was like a little room that the monks would live in the monastery. So he described what he was looking at as cells, and this is how we get the term that we use today. So the small building blocks of life are called cells because Robert Hooke used that term to describe what he was seeing. Now, more than 2 centuries passed before significant new discoveries were made. Now, it's not that scientists weren't studying cells, and it's not that scientists weren't interested in what was going on in the microscopic world. They were just limited by the technology that they had. So in the early 1800s, new techniques in making lenses came about. So with better lenses come better microscopes. With better microscopes, we can make more discoveries. So here we have two scientists that were contemporaries of each other, meaning that they both lived and worked in about the same place, and they often collaborated on their discoveries. So at the top here we have Matthias Schleiden. He was a German biologist, and he was looking at slices of plant tissue underneath the microscope. And he concluded that these cells that Robert Hooke had described something like 150 to 200 years earlier was the building block of plants. So Schleiden is credited with the conclusion that plants are made out of cells. At about the same time, another German scientist, Theodor Schwann, was also looking at things underneath his microscope. He was looking specifically at animal tissues. And again, through his investigation, he concluded that animals are also made out of cells. So with these two scientists, we get a very important conclusion that all plants and all animals are made out of cells. Now at the time, in the 1800s, this was all they thought there was, plants and animals. So what they concluded is that all life is made out of cells. Despite the fact that we know more today than what Schleiden and Schwann knew during their day, their conclusion has proven to be correct in that all life is made out of cells. The next scientist is another German scientist by the name of Rudolf Virchow. Virchow studied the works of Schleiden and Schwann and basically came to a conclusion. This was something that scientists had kind of thought about for a long time, but now they had gathered enough evidence to say, since all living things are made out of cells, new cells have to come from pre-existing cells. That is, cells don't just like pop out of nowhere. Cells have to be born from other cells that are already alive. So living things must come from other living things. This goes contrary to a common conception during this time, something called spontaneous generation. It was the idea that these vital forces could come together and life could just appear out of nowhere. Well, Virchow showed through his experimentation that life has to come from other life. The work of all these scientists and more allowed us to put together a cohesive idea of what cells are like. And this is what we call the cell theory. And there are three main points to the cell theory. The first point is that all living things, plant, animal, fungus, bacteria, everything that is alive is made out of cells. The second point is that cells are the smallest working unit of life, regardless of whether or not you are made out of a single cell or many, many cells. The cell is the smallest part of you that can still be considered alive. And then the final factor is that all cells come from pre-existing cells through a process called cell division. Scientists have observed this process of cell division, and we'll talk about this in more detail a little bit later on in the year. But what this basically says is for one cell to make a new cell, it essentially has to split itself apart. So now where we once had a single cell, we now have two cells. And this process can go on and on and on until you have literally billions and billions of cells. Now, the final detail we want to look at here is something that we alluded to when we were talking about taxonomy. When we break down and classify living things, the first level of taxonomy is the domain. One of the fundamental factors in each domain is the type of cell that the organisms that belong to that domain possess. And there are two main kinds of cells, what we call the prokaryotes and the eukaryotes. The prokaryotes are the smallest, simplest kinds of cells, and they lack a nucleus on the inside and any other membrane-bound structures. So a prokaryote is about as small and as simple as anything can get and still have what it takes to be a living cell. We'll talk about this in a little bit more detail in Unit 3, but the prokaryote basically has a membrane on the outside. It's filled with fluid and has a few little bits and pieces, but not much else. So prokaryotes are the simplest thing that can still be considered a living cell. There are two domains where we find prokaryotes, domain bacteria and domain archaea. These two domains are populated exclusively by prokaryote cells. The other kind of cell is what we call the eukaryote cell. These are larger, more complex cells. These are cells that contain their genetic material, aka their DNA, inside of a membrane called the nucleus. So when asked what's the difference between a prokaryote and a eukaryote, the most common answer they're looking for is that prokaryotes do not have a nucleus and eukaryotes do have a nucleus. Now, there are some other significant differences, such as that eukaryotes have more internal compartments and they have more parts inside of those cells. So when you typically look at a diagram of a cell, like the one in purple on this slide, you see a lot of bits and pieces on the inside of the cell. These are called organelles. These are the parts that make the cell function. Well, these are more complicated structures that are only found in eukaryote cells. Domain Eukarya is where we find organisms that have eukaryotic cells. Looking back at our taxonomy, there are 4 kingdoms that belong to domain Eukarya. Kingdom Protista, where we find small single-celled organisms that are eukaryotic. Kingdom Fungi, where we find the fungus. Kingdom Plantae, of course, is where we find plants. And Kingdom Animalia, where we find the animals. You, as an animal, are a eukaryote organism. So all your cells have a nucleus inside of them, and they have complex cellular structure.